All right, good evening. What a crowd. Very exciting to see a diverse crowd this fine evening. I'm Gokul. I work for the archives. I look after the public engagement programs at archives. A lot of familiar faces, a lot of new faces. So I'm going to quickly introduce you all to archives at NCBS. We're a public space collecting materials related to contemporary science in India. We have over 400,000 different items relating to 60 plus collections, which are housing in our archives, which is in the basement of the ELC building at NCBS. So we do a lot of public engagement programs, part of which is Archives Public Lecture Series, which we host every month, where we invite an artist, a scientist, a historian, to come and talk about their work and the approach that they take to their work. And today I am delighted to welcome you all to the 72nd edition of Archives Public Lecture Series, where we also have a collection launch of Krishna Bahadur and yes, Ranganayaki. So there's an exciting lineup of events. So to quickly give you what's happening this evening, we're going to start with Professor Shashi Thutupalli, who's going to introduce the speaker of the day, and then Professor Nanjundiah is going to talk about the origin of life. And then we have Parvati, our archivist, who's going to talk about the collection that she processed, that we processed at the archives, followed by Shashi Thutupalli's talk, Professor Shashi's talk on the scientific journey of Jeewanu. And we also have a couple of students of Krishna Bahadur and Ranganayaki who are going to come share their experiences this fine evening. Thank you.
So welcome to NCBS. It's amazing, this gathering I had never come across so far in my life. It's not about just in NCBS, anywhere. The kind of galaxy of intellectuals here today is, I think, take lots of photographs of this audience, please. And I don't know whether, when will be the next one of this kind. We have biologists, physicists, mathematicians, geologists, geophysicists, an artist, and novelist, and writers, and so forth. So very quickly, those who are coming for the first time, please do come again, and more leisurely you can walk around the campus and also understand what we do here. National Center for Biological Sciences, as the name indicates, is a research institution in the area of biological sciences, but across scales. We have people working at the atomic scale all the way to forest ecosystem, and not only the size of scale, and also time and complexity scale. And very accomplished place, well funded and well equipped for research in the modern area of biological sciences. In addition, we have a very large program of outreach, education outreach, public engagement, particularly in the area of science. Archives is one of those such organization in which we have a collection of some of the fantastic scientists of the last 100+ years who have contributed immensely to the growth of science in the country and in the world at large, including people like Swaminathan and and many more, right? Krishna and others. And today we are going to launch one such collections who have basically pioneered in the world in the work of biological sciences from the very origin of life, and which is very important, and Vidya is going to talk about. Before I introduce Vidya, I thought I'd just introduce the topic to some of these people, just to steal some of Vidya's time. Can I have the next slide, please? So all of you, those who have not come from biology background or science background, I'm sure you all have asked this question about what is life. But the science has brought a lot of answers to this question in the last 150 years or so. In that 150 years of science, in the next slide, has sort of brought deeper insight, next slide, into what's called chemistry of life. And across all forms of life that is on Earth today, and or anything that existed as far as fossil records tell us, they have exactly the same chemistry of life, right? DNA is the genetic material, how information is coded and decoded and so forth. And one major important aspect of life in the next slide, amongst the many things we know about life, from physics, chemistry, mathematics, and biology is information transfer fidelity. At the same time, there are certain variations are added. Otherwise, life would not be able to adapt to changing environment. And environment change is constant. It was something which changes even if there is no biological life, and also changes because there is biological life, right? For example, oxygen in the atmosphere is the product of activity of life. So given this, we have much, much deeper understanding of life. But there are still many, many areas which need deeper studies, which are less known. One of them is origin of life, amongst the many. The next slide, please. So next slide. And origin of life question, amongst many people, Darwin has also asked this question. In the last paragraph of his book, Origin of Species, and that's what he has written, because it's a deductive logic of a tree of life, he predicted there could be a single origin, right? And which has proven now, because all known biological systems have the same chemistry of life, so there must be a single origin. And that's the sort of search that's going on for the last maybe 70 years now, since the DNA double helix and the genetic code. And in that 70 years, many people have contributed. And one of them, of course, is the famous scientist from Allahabad, and which Shashi is going to talk about at length, and somewhat forgotten story until now. And we're going to launch the archives and then going to talk about more about it. And I'm not going to reveal anything about it because Shashi is going to talk about it. I'm not going to talk anything about origin of like Vidya is going to talk about. So to introduce Vidyanand Nanjundiah, a philosopher of science, historian of science, and a scientist, both experimental, conceptual theory, and as well as the mathematics and physics integration in his science. He has worked on self-organization. He has worked on developmental biology, and also a fantastic teacher. And he has taught in many, many courses, both in formal and informal setup. And he's been invited all the time whenever there is a complex question that we need to ask, and how to even start asking that question. That's where we always ask Vidya. So in a way, origin of any research, if we had to start, we generally go to Vidya and said, how to ask this question, where do we begin with? And today, before we launch the work of Krishna, Bahadur, and Ranganayaki, we're going to ask Vidya to introduce the topic and where the world is in this area and what should be the way we should be going to. Vidya, over to you. Thank you.
Thank you very much for those nice words of introduction, Shashi. And thank you too, Shashi, for having invited me here to this function. This topic is a fascinating one. One can spend endless time on it. I am not an expert in this area by any means, but I suspect, like everyone else in this audience, have been nagged by the question of how it all started at some time or the other. So what I'm going to present to you is my best understanding of where the field is today as developed over the years by people who have actually worked in the area. And I've titled my question purposely, how did life originate on Earth? And have presented in front of you now 5 themes which will take us to the end of this talk. I begin with asking the question that Shashi did just now, what is life? Then why do we think that it had an origin at all? And since the title of the talk says, how did life originate on Earth? When did it come to Earth? Then, of course, the most intriguing question of all, as well as the most difficult one to answer, and I may as well tell you straight away that we don't have an answer, but nonetheless, that doesn't stop us from asking the question, and hasn't stopped many people, including the people we are honoring today, hasn't stopped many people from trying to look into possible answers. So how may life have begun? And interestingly, there have been both worthwhile theoretical as well as experimental contributions. Then I sum up at the end briefly and offer a few reflections on my own on somewhat general things. What is life? Well, one can go into great detail about defining its properties. I'll let you read what's there on the screen. Basically, there are two properties of life that seem to sum it up. One can quibble over things, one can talk about things that are neither here nor there, at the boundary and so forth. But in general, life is an organized form of matter which can maintain itself, part one, maintain itself, which means try to keep at bay the inevitable decay due to thermodynamic effects. And in addition to maintaining itself, it can make copies of itself in a certain sense. So these are two aspects of life that are referred to as metabolism and reproduction. And for the moment, I'll stick to these two aspects. Then, of course, there are many other things which you see in the fine print there, so to speak, on that slide. Metabolism and reproduction. And we'll stick to these two, as I said, as our defining elements for life. Most importantly, of course, we need to keep in mind that life is a property of matter. And therefore, we rule out automatically any explanations of life, of the nature of life, and so forth, which have other ways of posing and answering questions, philosophical, religious, etc., etc., etc. So for our purposes, life is a property of matter, and we study life in the way we study any other kind of matter. Right. Now, how many forms of life are there? This has traditionally been answered in forms of what are called counting what are called species of different sorts. But this is an interesting way of categorizing life that came out not very long ago, which is into plants, bacteria, animals, single-celled creatures of other kinds called protists, viruses, and so forth. But it categorizes them according to what is called biomass. In other words, how much do they weigh, totally speaking. And you take that as a measure, some measure of how successful they have been on this earth, perhaps, how successfully they have propagated. And these weights are given in gigatons, which is to say 1,000 million tons of carbon. The element carbon turns out to be an essential component of all forms of life. And measuring weights in this way enables you to avoid looking at forms of life which are sometimes hydrated, sometimes dehydrated, and so forth. So in terms of gigatons of carbon, plants are the most common form of life. I'm asking you now to look at the left part of this slide. Plants are the most common form of the life. Bacteria are second most. Then there are these things which are really part of our ancestry called archaea. For the moment, you can think of them as bacteria of some kind, which are another form, etc., etc. There are fungi. Animals to which we belong are a very tiny part of the whole thing. So if you look at the whole, it comes to something like a few, 1% or 2% of biomass on Earth. Fine. Now, if you expand that animal triangle which you see on the lower right of the left panel and blow it up, there it turns out that the most common animals, again measured in these units that I described to you, are insects and their relatives, what are called arthropods. They're really overwhelmingly dominant. Then you have fish, then you have roundworms, then you have things like hydra and so on, you have mollusks, they're pretty common. Humans are a very, very tiny fraction of this. 0.06 out of the whole thing taken together, which is 2. So again, you can see that it's about whatever, a fraction, a small percentage, not at all big, 2-3%. Now, where are these forms of life found? They're found on Earth. To date, there's no form of life that we know of which exists outside Earth. And here on Earth, it turns out that most of the weight carried by life in the same units that I told you earlier is on land. So most life is terrestrial. Again, I ask you request you to look at the left panel here. Most life is terrestrial. There's some which is marine. Interestingly, there's a substantial component which is in what's called the deep subsurface, which really means a kilometer or a couple of kilometers. People have gone up to 4 or so kilometers in South African gold and diamond mines. You also find life forms there, and added up, it's estimated that their biomass accounts for quite a bit. Okay, out of these things, the classification that we made earlier, how much do you find on land and how much do you find in water, in the seas? This is the right-hand side of the picture. Plants are almost entirely on land. Fungi are almost entirely on land, but you also find them in water. But then when you come to single-celled creatures, animals, bacteria, and so forth, the balance slightly begins to shift. And with microorganisms, bacteria and archaea, microorganisms of this type, bacteria and archaea, they're overwhelmingly in water. So their contribution to the land fauna is minimal. If you look at the elemental composition of life, if you look at the standard elements in the periodic table and ask how many of them are found in life, even a quick glance across this table where humans are taken here as representatives of living forms, not entirely accurately, but that's okay. Plants would give you different percentages. One thing that you see immediately is that the relative representation of these different elements, these are all figures given in percentages, these relative representations are very, very different between the human body, which we have taken as here standing for life, the ocean and the Earth's crust. You can push this a bit deeper, but it's fairly clear that living forms are not in any way statistically fair samples of what we have around us. So there's something very special which has been brought together. So that's so much for what life is. Why do we think it had an origin at all? After all, it could have been that whatever life forms you see now have been there all along and will continue to be this way all along, for all you know. So why talk of origins? Well, many reasons for doing so. One, it was appreciated long ago that both in terms of metabolism, which is represented here by a bacterium, and in terms of their other properties, which you can generally call genetic properties, there's a huge amount of similarity between living forms. So in essence, pared down to their basics, so to speak, all living forms are the same. You have to say this with some amount of hedging, but by and large it's true that they're identical in terms of their internal metabolism and in terms of whatever they do to replicate themselves. So there are these quotes. Monod's quote has become more famous. Anything that's true of E. coli, which is the name of a bacterium, must be true of elephants, except more so. You have to decide what that means. But it turns out that Cloyver, who was a distinguished bacteriologist and microbiologist, said the same thing earlier in terms of biochemistry. Now, in terms of what you might call the mechanics of reproduction, there are two elements that tie together all life forms. One is the unfortunately named central dogma, but that's a name we have come to live with. And what that central dogma says is that there is something called DNA, something called RNA, something called protein, and in general, again, a word which is a phrase which is not very easy to define, but I'll use it nonetheless. In general, the transfer of information transfer, moves from DNA to RNA to protein, sometimes from RNA to DNA, but never from protein to either of these two. So there's some asymmetry in information transfer in living forms from these molecules on the left side of the figure, what are called nucleic acids, to the molecule illustrated on the right side of the figure, what's called protein. Now, the important thing to remember here is that nucleic acids and proteins seem to be the two kinds of molecules which are central to living forms. There are also rather complex sugars, but these sugars are usually products of things done by proteins, enzymatic action done by proteins on things that creatures eat, that we eat. So in general, it's the nucleic acids and proteins that are believed to hold the pride of place. Then, in addition to that central dogma, there's something called the genetic code, which is also a very curious entity. It is a set of formal relationships. I repeat the word formal, as written down. It's a set of formal relationships between the DNA or RNA that you see in this picture and the proteins. So it tells you how these molecules on the left, which I just now told you a moment ago, transfer information to proteins, convey the information that is used to make proteins. The genetic code tells you precisely what underlies that information transfer, and it turns out that there are bits and pieces of this DNA, what are called triplet codons, triplet nucleotides which constitute a codon, which are in correspondence, one-to-one correspondence with a certain number of basic building block entities of proteins that we call amino acids. Now, the correspondence is one-to-one in one direction, but not in the other direction. There are interesting questions related to that I might mention later. But the point for our talk is that both the central dogma and the genetic code apply across life. So they are the things that, so to speak, are the driving engines behind everything. Now, I have to jump a bit in terms of the chronology of this talk. The other reason for thinking that life had an origin is really pretty recent. And it dates by and large from the time of Darwin and Wallace's elucidation of natural selection, which is now 150 plus years ago. And it is based on the fact that when you classify living forms, when you categorize living forms, they seem to naturally fall into a tree-like structure. Now, when I say naturally fall, obviously a lot of questions spring up in your minds immediately. But for the moment, please take it for granted that you can arrange the living forms which are on Earth today. Please remember, we are only speaking of forms that are around today. We can arrange them in a way that they are connected in this fashion that you see illustrated on the screen, so that any two living forms today can be hypothesized to have had a common ancestor if you go back sufficiently in time. And if you go back far enough, everyone had one ancestor. So basically what I'm trying to tell you here is that the most economical description of the diversity of life on Earth today is that this diversity is based on a similarity because of descent, because of a common origin. So this is a you might almost call it a logical proposition, which you can obviously accept or reject, but by and large it turns out to be a valid proposition. There are some caveats that we'll see later. That particular proposition has been strengthened with the coming into age of molecular biology. Now all these words that you see under bacteria, archaea and eukaryota, the words in smaller print below them, they're exactly the equivalent of these plants and animals and microbes that you saw sketched in this cartoon here, except that these are specific names, actually generic names that are given here. So it turns out that you can perform the same exercise of tree building now with the help of DNA sequences that you extract from all these organisms, and then show that the most economical way of understanding the distribution of these sequences is in terms of common ancestry. Once again goes back to a hypothetical common ancestor, and that common ancestor came to be called, and is still called for that matter, LUCA, for last universal common ancestor. But once again, the existence of that ancestor is a deduction based on analysis. It's not something that we have evidence for, but it seems the best possible conclusion you can draw based on this evidence. By the way, the tree itself is interesting. On the left-hand side, you see one big branch at the beginning going off into the bacteria. The right branch is interesting because it doesn't come to us. We come under that thing called eukaryota at the extreme right. And you can see immediately that we share a closer relationship with something called archaea, a name that you saw before, a much closer relationship than we do with the bacteria. The surprising thing here is that the archaea, in a sense, look and feel and smell and so forth like bacteria. So we've had a closer bacterian cousin than we usually think of. So it's not going all the way in the back. Okay. So we've made 2 points now. Try to get some idea of what we mean by life, and then try to make it plausible that life had an origin. Now, of course, we have to figure out how it came to Earth. Quick look at the Earth here, and for our purposes, the salient points about this figure is that the inner core of the Earth is solid, the innermost core is solid, then there's a liquid outer core, then there's something called a mantle, which is in fairly violent activity. At least the upper part of the mantle is in violent activity. On top of that, there's the crust, which is where everything that we call our world exists. So this is 0 to 40 kilometers according to this slide. So our world, for most practical purposes, ends at 40 kilometers below the surface. The Earth itself, of course, has a diameter of 6,000, radius of 6,000 plus kilometers. The first 40 kilometers of that are all that matter for us most of the time. Interestingly, the mantle, where there's a lot of activity that is going on, also turns out to play a role in the story that we are trying to decipher here. History of the Earth, again, I'm not going to go through this in detail. You can look at the slide. Basically, the idea is that this developed by budding out from some cloud-like object, which also gave rise to the sun as at its remnant, as a remnant, and the other buds became various planets. The interesting thing is, we can say when this happened, or the geologists, the geophysicists can tell us when this happened. This is estimated to be about 4.5 billion years ago. And once it was formed, many other things happened. The moon budded off from the Earth, probably by a collision from something else. And there was a lot of activity going on in the Earth. A solid crust developed after some time, what we call land. And most importantly, liquid water came onto the Earth as a consequence of standard geological, geophysical processes. And there was no oxygen. So this absence of oxygen also turns out to be an important clue in developing this story. So you had a reducing atmosphere and liquid water. There were two other things that turn out to be germane, but I won't go into it in details here, which is this activity in the mantle, which has led to what's called tectonic activity in the crust. Turns out to have played a role, perhaps. And the second thing which is very interesting is the stabilization of the Earth's magnetic field, which happened about 1 billion years, it's believed, after the Earth was formed. Also turns out to have had a role, but I won't go into that. Ancient atmospheres, I've told you this already, hydrogen and helium were both light enough to escape. Secondary gaseous atmospheric products developed because of volcanoes, outgassing from the surface of the Earth, and so on and so forth. Oxygen itself, interestingly, almost certainly, it's believed, had a biological origin, not a purely geological origin. So you needed living forms first to make oxygen, photosynthesize, as we know, and then, of course, release it and give it into the atmosphere. So this is a slide which tells you in this circular coordinate what events occurred during the life of the Earth, and what were the geological phases into which the life of the Earth has been divided by people who work in the area. The only reason I'm showing this slide, one can't see it in detail, is that if you look at 12 o'clock, which is right at the top, which is essentially the time when the Earth was born, so as I said, roughly 4.5 billion years ago, and come all the way around, so from midnight, one night if you wish, to midnight the next night, the appearance of life on Earth was very, very recent. You can already see that, okay, somewhere around 10:30. And the appearance of humans, of course, was even more recent, somewhere close to midnight itself. You can't resolve it on this slide. So we have an interesting phenomenon here of something which was the product of events on Earth, which appeared very, very, very recently, has barely made its presence felt in terms of time, in terms of occupancy. Now it's trying to speculate on what happened in the past and what led into its own being. Yeah. So various things, I've been through some of these. The Earth was formed, there were volcanoes, there were earthquakes, plate movements. Cosmic dust, this has been thought to be a very important element, or at least an important element in the formation of life on Earth. The magnetic field I mentioned, and then finally prebiotic chemistry, on which we will spend some time. Okay. Now, so life had an origin, it came on Earth, and the Earth itself has been in existence 4.5 billion years, so the life on Earth that you see now can't have had an older existence. Indeed, it can't, logically speaking. And the best evidence for life that we have, amazingly enough, from a kind of fossil left behind by bacteria. It's not really a fossil, it's an extracellular exudate of bacteria which accumulates, things called stromatolites, have been dated to about 3.5 billion years ago. Now there are other signs which indicate that life may have actually appeared even 4 billion years ago, but that's the range of time that one is thinking of. And these forms of life are already advanced forms of life. These are bacteria-like creatures, we presume, that were responsible for these layered mat-like entities which are called stromatolites. So the actual beginning of life, of course, must have been much before that. These are fossils which come from an ancestry similar to ours, 1,000 to 1,500 million years ago, 1 to 1.5 billion years ago. And yet the oldest fossils of this kind have been estimated to be 2.5 to 3 billion years ago. This is work which actually shows you what people see, which they interpret as evidences of primeval life. So this is from something called the Pilbara crater, fraction of the Earth's crust that you have access to in Western Australia. So these are images of things, of structures that are seen there, within which it's inferred that you have forms of organization that can best be interpreted as cellular material, that is some form of living material. And in fact, again, to jump ahead with the story very much, this sort of study into living forms which seem to mimic what may have been in that ancient earth, which was extremely hot, did not have oxygen, right, and had a lot of tectonic activity. So the living forms that you have on earth today, which mimic those ancient living forms, can all be grouped in a certain way, which gives rise to a tree-like structure of the form that I've shown before. And that tree goes back even further. So it's hard to say exactly how much. Some people say as close to the origin of the earth itself as a few 100 million years. But that tree also is based, as I said, on advanced molecular biology techniques. So all the creatures that you see on the top of this tree are living forms, exactly as they were in the other trees that I showed you. But these are living forms which seem, in many ways, to be extreme forms of matter, what are called extremophiles sometimes. They like very high temperatures, they like very high pressures, things of that kind. And they seem to group in this way. So again, tells you the root probably goes back very close to when the earth itself was formed. Okay, so rough kind of thing. We don't have any clear idea of how life came on earth, but we know that it came on earth, and we know that it came certainly by a certain time period. Now, how did it begin? This, of course, is the subject matter of our talk, and this is where our ignorance is maximal. But as I told you, there are interesting theories and experiments that go into this thing. One kind of speculation that has held ground for a long time is that life was seeded from outer space. This goes back to the Greek philosopher Anaxagoras and comes to as recent times as Crick and Hoyle and Vikramasinghe, all of whom advocated this reasonably seriously. The main reason behind the more recent supporters of this theory for the origin of life, sometimes also called panspermia, so seeds being dropped from outer space, so to speak, figuratively speaking. The main reason for their supporting this sort of a theory is because they thought that the available geological processes on Earth, which could presumably give rise to interesting things happening, we'll see some of what those interesting things are, they wouldn't have had enough time to do their job within this small window of, what is it, a few 100 million years within which life seems to have arisen and given rise to cells. So it's not just some kind of origin of life, but origin of cellular life. So all of them seem to have felt, or at least convinced themselves, that there wasn't enough time to do these things, in which case why not take help from the rest of the universe? And the universe, of course, has been around very much longer than the Earth, and that could be a plus. Some reasons which also fostered this belief is that meteorites, which have impacted the Earth, and which have clearly come from away, certainly from outside the solar system, and sometimes from even much greater distances away. Meteorites have interesting molecules in them which give you hints of what you might call organic molecules. Some of them are indeed organic molecules. Formaldehyde has been discovered, for example. And so there's a clue coming from there. Right. Now I'm going to go very rapidly through the kinds of thinking that people have presented to try and explain how life may have first originated. Chronologically, Alexander Oparin comes first, and his idea was that essentially you had the right temperature, you had the right ingredients, right, which were needed for two things to happen. What are these two things? One was to close off an inside from an outside. In other words, to partition something into a small region which was enclosed, and the rest of the world which was outside it, what you would today call a cell. So spontaneous self-assembly of molecular entities giving rise to some sorts of protocells. So these were droplets. Sometimes Oparin thought of these not necessarily as droplets with what you might call a hole inside them, as you tend to think of cells usually, but solid droplets, nonetheless coherent solid droplets which were distinguished by having a surface boundary, which were distinguished from the outside world, and because they were coherent, things could happen inside them, okay, because they were small and So he called them co-acervates, so they could assimilate things, put things out and so on. As we will see, the idea has been developed for that. The same idea, amazingly enough, was put forward 5 years later by J.B.S. Haldane, and Haldane was a great coiner of words and phrases in addition to many other things. And he said this primeval medium within which this kind of condensation happened, which enabled some sort of primitive cell-like entity to separate from everything else. This primary medium was a hot dilute soup. So that phrase remains in people's memory even if the details of what Haldane said are forgotten. And the hot dilute soup essentially, as I said, is the same thing that Oparin had proposed. And there's an interesting small story here which I'll risk telling at the cost of some time, which is Haldane didn't know that Oparin had done this. Oparin's work had been published in Russian, and Haldane had no access to the Russian literature then. So he did this independently. Later, of course, both of them realized that they were two people who had the same ideas. Now, Oparin's own finding came about in the following way. Oparin joined someone to do a PhD thesis. Those of you who are students of evolution here will know the name, a person called Timiryazev, Clement Timiryazev, a very distinguished Russian, pre-Soviet Russian botanist, who was among the staunchest defenders of Darwin at a time when Darwin's ideas were being subject to a lot of flack, shall we say. So that's what Timiryazev. Oparin meant to do his PhD with Timiryazev, and Oparin already had these ideas of origin of life and so on and wanted to work on them. So Timiryazev apparently took him on and said, well, this doesn't look a feasible PhD project to me, but go ahead, I'll give you 3 months and then you come back and show me life, essentially. I'm not quoting him, of course. Incredibly, Oparin came up with something which these were not experiments, so he couldn't show him anything which was moving or kicking, but came up with interesting ideas which Timiryazev thought were worth following up. But even then, Oparin was unable to publish his findings because this was the time when Russia was in ferment, so this was the immediate post-revolution years. It was impossible to find a publisher, which was lucky for him because many of his ideas turned out to be wrong in terms of the things that he had. So the delayed publication helped him, as I think some of us will agree has helped us too occasionally, but we keep complaining about it. Hot dilute soup. Okay, now very rapid. And we'll look at two of these at least in some. Miller and Urey came up in 1953 with an experiment which has become famous, which is really the same as the coacervate experiment or Haldane's proposal, coacervate or Haldane proposal, except that this is an actual experimental test. So far we have just been dealing with theoretical proposals. And this was to flash lightning, a source of energy, which was really an electrical spark, you couldn't have lightning there, inside a beaker which contained methane, water vapor, and ammonia. So the kinds of things that you thought were in the primitive atmosphere. And lo and behold, this experiment went on for many weeks. Lo and behold, what emerged at the end was a lot of messy stuff on the walls of the beaker, as you might expect. But some of it was organic molecules. So here was a way in which, remember the name of the game now, prebiotic chemistry I said, so the name of the game now is to move from inorganic chemistry, so had been going along all the time, of course, ever since the universe was created probably. But the aim is to move from prebiotic inorganic chemistry to prebiotic organic chemistry. So this was one way in which this could happen, said Miller, who interestingly also did this for his PhD thesis with Urey. Then Juan Oro did something else which was very interesting too in its own way. He managed to synthesize in the lab one elementary units that go up to make up the DNA molecule, not strictly the unit itself, but adenine, which is the base which goes into that unit. 1961. Sutherland very recently went further than this and actually synthesized that managed to synthesize that entire unit in the lab. And it's of some interest that the starting material for both of their experiments was a cyanide of one kind or the other. One used hydrogen cyanide, which we all know is not good for the health, but from it you can get life, and maybe you needed it to get life. Bahadur and Ranganayaki, which is the reason we are all gathered here, spectacular experiments. Again, the same kind of thing, but you're going to go into the experiments in detail, so I won't dwell on them here at all. Fox and Harada, at about the same time, but slightly later than Bahadur and Ranganayaki, also repeated this kind of thing, right? Trying to get material in the lab to spontaneously condense into some kind of a form, which, remember, the goal is entirely to isolate yourself from the rest of the universe, right? And within yourself to have chemistry of an interesting sort going on, because components which have become isolated. And so that's an illustration of the Miller experiment. You can see lots of beakers, and there's simulated lightning at least, and there's rain as well. This is a cartoon, obviously, but the real apparatus doesn't look very different from this. And then you have a condenser where everything comes down, and that's where the products were collected. Ranganayaki and Krishna Bahadur, again, I just mentioned their names. I have a photo later, thanks to Shashi, but we'll hear much more of them later. And I think these are really inspiring words. Since the 1950s, for more than 30 years, we, together with our research students, have been investigating one or the other aspect of origin of life. More of this to come. Right. What did they do? One big difference between everything that went so far and what they did was that they managed to get their energy source directly from the sun, as indeed we do now. So the source of energy in many of their experiments was light, visible light, and they managed to show that these things that they created, we'll see what they were, carried out photosynthesis, which means they were able to carry out these forms of primitive chemical assemblages which managed to grow, which managed to divide, which managed to metabolize, also managed to assimilate free energy. Slide from Oro's experiments is just for fun. As hydrogen cyanide goes down, adenine goes up. I told you this already. Sidney Fox, proteinoid. Fox said something else which was interesting though. He said that these proteinoid-like forms can also be found in nature in some places, including lava from volcanoes. So, which suggests that something like what you are trying to hypothesize may actually be taking place even now in their living condition. So, these are pictures of these microspheric proteinoids. You will see very, I think, more impressive pictures later from the Ranganayaki Bahadur experiments. One more point before I end this part of how it could all have started. There's a very interesting proposal not long ago, the data you see is 2016, a very interesting proposal for how purely chemically, right, you can get this sealing off between outside and inside. That you knew, of course, already. Many people had shown that. But in addition with this sealing, given the right sorts of ingredients there, you can set up gradients, electrical potential gradients, which again immediately can be used as in a battery for driving a current and doing useful work for biosynthesis. Just one word about all of this. I've already mentioned DNA, RNA, and proteins, and I've said information transfer is from DNA to proteins, or from RNA to proteins, not back from proteins in any way, to either DNA or RNA. So on the surface, it might seem that DNA and RNA are on a par, but in point of fact, it turns out that RNA is much more fundamental than DNA in terms of being simpler in terms of its properties, right? And also more sophisticated in terms of many of the things it can do that DNA cannot do, at least so far to our knowledge. So the belief has grown that even though the chain of information transfer today suggests DNA and RNA on the left and proteins on the right, perhaps it began with RNA giving over to something like protein, and DNA came later on the scene. But this is certainly very much later than the time at which, about which we are trying to speculate, which is the very origin of living matter. And almost certainly that couldn't have had anything to do with either DNA or RNA, both of which are extremely complex molecules. Fine. So I'm through with the bulk of my talk now, in fact almost all of it. I'll sum up what I've said and then offer a couple of reflections of my own. Okay. Right. Very interestingly, the first scientific origin of speculations on the origin of life may well have been Darwin's, except that the first evidence we have of Darwin's thinking on this was to say that Darwin thought it was a waste of time to think of the origin. So this is in a letter to his friend Hooker. So he says it's mere rubbish thinking at present of origin of life. And this is a nice kind of almost ironical statement in the light of what's going on in science today. He says one might as well think of origin of matter. So trying to suggest, if you want to come up with an impossible problem, origin of life is on the same level as origin of matter. The world has turned around so much that a distinguished cosmologist, Susskind, not long ago said modern cosmology really began with Darwin and Wallace. In other words, trying to suggest that just as these two problems, the origin of life and origin of matter, seem to be similarly intractable at one time, right? And the reason they were intractable is, I hope, obvious by now. They dealt with events in the distant past about which you could speculate. They dealt with properties of creatures that you could only speculate about. I mean, the creatures about whom you could only speculate. And basically, these were historical questions with primary data, by and large, lacking. By the way, this may not be entirely true of astronomy, because in a sense you can look into the past. But in biology, you cannot. There is no way of looking into the distant past, but you can use inferential reasoning. So what is interesting now is that, of course, in both fields, by using very careful reasoning, logical reasoning, some experiments that you can carry out on Earth, right? And making sure that your logic holds together tightly with whatever you know about the laws of nature, about the workings of nature, try to build up a plausible picture, which you do in, one understands in cosmology as well. So he said, Susskind said recently, modern cosmology really began with Darwin and Wallace. But then Darwin wrote another letter to Hooker, 1871. So our chronology is a bit off here. Please notice 1863, 2008, and then 1871. This also finds a small kind of place in his Origin of Species. Darwin came around and said, no, hang on, I was very pessimistic at one time, but maybe things can happen, maybe. And look at his guess here. A warm little pond, ammonia, phosphoric salts, heat, electricity. Then there's something else he says after that. He says, look, with these kinds of things, maybe you could get some kind of life appearing. But then he says, if that happened today, and for all you know it is happening today, that would be promptly eaten up by the forms of life which you already have, by the more advanced form of life. So basically trying to say, any organic matter that is produced on Earth from inorganic matter on Earth will immediately be degraded. I mean, this happens all the time. In fact, not very different from the kinds of things that happen after death when life ceases. So Darwin said this could have happened, and so maybe, life won't be arising today anymore. So that was one part of my background. Secondly, we have to keep in mind that everything that we have been saying here, and indeed I would say everything that it's possible to say, has to be based on physics. And here I include chemistry as a part of physics, of course. So physics is, as someone has said, the only game in town. There's nothing else outside. The only alternative that I can see is black magic, and there are many people who believe in it, and good luck to them, and but it's not on the table as far as we are concerned. Okay. But at some stage in biology, a new principle comes into play. This principle has nothing to do with violating the laws of physics or being extra-physical, non-material, or anything of that kind. This is a principle called natural selection. This is the one which was first explained clearly by Darwin and Wallace. it's hard to describe it in a few words. Shashi gave an indication of that where he told you that living beings made copies of themselves, but the copies were not all the same. So natural selection builds on this statistical variation between things that must occur simply because of physics and chemistry. All chemical reactions are statistical reactions. So if you use chemistry to make something, whatever it be, that product cannot be 100% copy of the original. It may be a copy of the original 99.9% of the time, but 0.1% of the time you get a variation. So natural selection builds upon this variation in a very nice way to say how diversity can be generated. So in fact, even though physics was the only game in town, certainly until natural selection came on the scene, and even now is essentially the only game in town, this added principle of natural selection puts biology on a footing which is slightly different from that of the other natural sciences and gives it a claim to some sort of quasi-independence similar to that of physics. Keep this in mind. However, this probably does not in any way affect our reasoning until this inorganic chemistry to organic chemistry transition happened. But after that, probably there are good reasons for thinking this happened. This, suppose, goes into a deeper philosophical question, which is that by and large, and again I accept cosmological or astronomical arguments here, by and large, physical science is an ahistorical science. Certainly in the way it's practiced in the lab, it's an ahistoric science. You want to understand the properties of matter as they are in front of you in terms of things that are happening right there or things that you believe are happening right there. Whereas natural selection told us that biology at its deepest has to be understood as a historical phenomenon. So this leads to very interesting differences in the way biological structures are built up and physical structures are built up. Again, I won't go into this in detail, but I just put this in your mind. Keep in mind what I said earlier though, which is this is not to say that physics does not operate in biology. In fact, there's only physics, but this is a new organizing principle. And it's been an interesting question, much explored in recent years, I'd say in the last 20 to 30 years, of how much physics there is in biology and how much biology there is in biology. So it turns out to depend on the level at which you're asking the question, the detail in which you're looking at the answer and so on. And we have a very fine physics of life program in which NCBS, ICTS and some other organizations are participating, which are actually trying to see how far they can push the boundary between physics and biology. Okay, these are two audios which I will, beg your pardon, two videos which I will skip now, not needed. Exobiology, I come back to something that we started with, which is the possibility that life was seeded from outer space. As I told you, philosophically it's not very interesting if you simply push the problem back, you have to solve the same problem there. And in fact, distinguished paleontologist and evolutionist Gaylord Simpson said exobiology is by the way, I hope it's clear what exobiology means. Biology not necessarily confined to the earth, but biology somewhere else. So Simpson said exobiology is a field that has yet to demonstrate that its subject matter exists. Now, unfortunately, we are still in that stage today. A great many exoplanets have been discovered. I think the latest count is a few thousand, in fact, as many 5,000, 6,000. So basically, not even Earth-like, what are technically called planetary bodies, which are believed to be in orbit around star-like bodies. You can't even say they are sun-like stars in every case. But okay, good enough as it goes. But are the conditions there such as to permit life to evolve? And of course, science fiction writers even go further and say you may have advanced forms. And the proof that you have very intelligent forms of life somewhere else here is that they have not tried to contact us. So you can go into such interesting speculation. However, it is true that we are in communication with the rest of the universe, and that may be meaningful, not just now, but may have been very meaningful in the past in terms of what raw materials entered the earth, certainly form part of the crust, as we said, and went into this primitive chemistry that we thought of. Very interesting lab experiments are going on now. again, they have to begin with RNA. You can't go before that. We're still stuck at that. There is this big missing link still. But you can begin with small RNA molecules and get extraordinarily rich biology in the lab. So you can get the equivalent of predators and prey. You can get the equivalent of different molecules collaborating with each other to form a consortium, right? And then trying to compete with other consortia and so on and so forth. So you can get a spontaneous emergence of what's called complexity in a test tube, which is also intriguing. One final point here, which is, and this is somewhat dismaying in terms of clean theories, this tree of life that you saw illustrated more than once, which had a single root at the bottom, now turns out to be not that clear-cut a picture of the evolutionary tree. In fact, to some extent, even among living bacteria, but more so among those trees that I showed for molecular species a while ago, species defined in terms of the molecular constituents a while ago, it turns out that there seem to have been side-to-side exchanges of material. What material? The kinds of material that we're interested in, probably DNA and proteins. That, of course, converts the tree into a web and tells you that if this is true all the way down, this picture only tells you that this is probably true for cellular life, which is already fairly recent, everything considered. But if you go all the way down and it was still continues to be true, as is looking to be the case, maybe there was no last universal common ancestor. Maybe we are all products of a committee of some kind. Who knows? Have to wait and find out. Okay. Now, the very last slide, absolutely, and here are a few remarks. Now, we are all gathered here, of course, for the main event of the day, which is to follow this talk, which is to pay tribute to this marvelous work done by Ranganayaki and Krishna Bahadur, and we're going to learn more of it in detail later. That makes one think of what they did in the context of something that we talk about a lot, which is science, modern science in India. And it's as fine an example as I can think of path-breaking work, entirely original work, really creative work, having been done in India, of which we can and should be justly proud of. And I think this cannot be said often enough. At the same time, we cannot be unaware of the fact that in today's India, especially, a huge amount of nonsense is being said in the name of Indian science. Huge amount of nonsense. Now, my point is not simply to say that everyone knows this, but my point is to say that we as scientists really must speak up more often against this kind of thing. The nonsense is beyond belief. It goes to the extent of saying, well, all of you know this, we had invented heavier than air flying machines, we knew all about tissue transplantation, even organ transplantation, we knew about cloning, you name it. I mean, the list is endless. And it's, so my point is not just that it's nonsense. I'm sure the same kind of nonsense is being spoken by people in many places in the world. I don't think we are in any way especially culpable. But the problem with us is that it's being spoken by people in authority who should know better than to utter such things. And as I repeat, we as scientists are not standing up against this often enough. I think it really is our duty in whatever small way we can to point out that this isn't true. This isn't the way you look at the past. This isn't the way you do history, let alone history of science, by giving yourself false glory by addressing the past. And in fact, by doing things like that, saying things like that, we'll be doing violence precisely to the memory of people like Ranganayaki and Bahadur, who have blazed such an incredible trail for us to follow. So I want to end with this kind of sentiment, which is not strictly speaking part of my talk, but it's something that I think it's important we keep in mind in the context of Ranganayaki and Bahadur's work. Thank you.
Thank you, Vidya. Really, you summed up very well, both the science and the sentiments. So I don't know if we have time for questions. Yeah, of course. So before I want to reiterate, many people say life is singularity. Maybe it has happened once in the universe. Maybe you didn't touch upon that logic or argument. This audience is a singularity today given Bangalore traffic. I think that's what please come here and take pictures. Ravi, go to the stage and take the picture of the audience. Okay. Just two questions, I guess.
So I'll be very brief. Thanks very much. Beautiful talk. So this is spurred by your comment about the RNA perhaps having been the progenitor, in a sense, of this whole process. And your first comment about the two main characteristics of life as metabolism and replication, it made me wonder whether could these two processes have happened at different periods of time or at different stages in this historical evolution, and in some ways may even have been logically, independent kind of developments. So I wanted to hear your thoughts on that.
Okay. Answer to that is very true. That could indeed have happened. But the reason for this great excitement about the so-called RNA world is that some years ago it was discovered that RNA could do both things. In other words, RNA could act like a protein enzyme, right, as well as the carrier of information to make proteins. Because of lack of time, I didn't go into these experiments in detail, but what they involve is taking a small segment of an RNA molecule which can code for an enzyme, code for a protein, and in addition catalyze the making of itself, in other words, replicate itself within a single molecule. So starting with this single unchanging RNA species and relying solely on the properties of chemistry to make mistakes, you get a whole variety of species with different catalytic properties, with different replicative properties and so on, which build this community. So what you say is perfectly true. Metabolism and replication could, in principle, have arisen at different times and then come together. The reason for thinking of both having come together and work together is because you have here a candidate who can do both things.
Yeah, one last question.
Yeah. So thank you, Vidya. That was a wonderful talk. Now you mentioned something right at the end, which is the context for so you talked about horizontal gene transfer and how that affects this sort of picture of the tree of life. And of course, you mentioned at the very beginning, you mentioned different possible origin scenarios, but in principle, one could conceive of different possible origins taking place together.
Yes.
Right? So I want you to say a little bit more about how, when do we have a situation where you don't have a tree of life? What are the interesting alternative pictures like this one that one should be thinking about in talking about
I'm
this?
not sure I follow your question, but I want to say one thing, which is, oops. Okay, could have worked with that as well. Okay, we'll work with that. By and large, this picture is valid, right? So these examples of horizontal transfer or horizontal exchanges of genetic material, they're pretty prevalent in bacteria. Don't happen very often, but pretty prevalent in bacteria. So far unknown in the lineage that includes us, or probably known only rarely via viruses, etc. So by and large, biologists still work with this picture. And in fact, there's a great deal of molecular biology in the marketplace which would go out of business if this was not true. Because much of what goes on now, what is called bioinformatics, depends on building evolutionary trees of ancestor-descendant relationships, assuming you have a tree-like topology. So that kind of thing you have to take care of. Otherwise, by and large, this is correct. Okay? But the horizontal transfer business in some cases, it's quite obviously led to a dramatic outcome. So, for example, the things that produce energy inside our cells, what are called mitochondria, it's fairly clear that they had a bacteria-like origin. In fact, we ourselves are in that sense conglomerates created when a member of one lineage, which is the bacterial lineage, fused it with a member of another lineage, which now you call the archaeal lineage, and gave rise to our lineage. So horizontal transfers in those cases have been extraordinarily important, otherwise the whole lineage would have been missing. But otherwise one works with the assumption that so far that they're not too serious.
Yeah, I'll ask you more later.
Okay, in the interest of the time, we'll have a 15 minutes tea break now, followed by launch of the papers of Krishna, Bahadur and Ranganayaki, and followed by Shashi's talk. This origin of life question is being asked for quite some time now, but unfortunately not many people are working on this in this area, so it's less known. And also people have to be very brave and adventurous to work in the area of origin of life. Shashi is one of them here. There are a couple of others who are working in India. Sudha Rajamani is from ISA Pune, Sandeep Ameta from Ashoka. I happen to be associated with all these three institutions working on origin of life. And Vidya will also be there. I think outside the tea time, you can interact with the people working and asking questions in the area of origin of life, and then come back in 15 minutes. Thank you very much.
Meanwhile, just out of curiosity, how many of you have been to archives before? Okay. How many of you have know what kind of work we do at the archives? Okay. How many of you found it really difficult to find archives at NCBS? Shashi. So clearly, there's not many hands up in the air. I think there's something preventing a lot of people, especially the general public, from accessing archives at NCBS, even though it's a public space in a beautiful campus at NCBS. So this is something that we are trying to address and figure out what's happening. So this is a start to our understanding of what's preventing people from accessing archives at NCBS. So it's a very, very short survey. I know people hate taking surveys. We came down from 20 questions to 3 questions. It takes 2 ticks and a sentence or 2 or 3 sentences. So feel free to do this when you're leaving outside. So it's just going to be right outside. It'll just take a minute, but it'll make a lot of difference in the way we address the issue of accessibility at archives. Thank you. So with that, over to Parvati.
Hi everyone, I'm Parvati. I'm here on behalf of the archives at NCBS. We welcome you today to a very happy occasion, the launch of the Krishna, Bahadur and S. Ranganayaki papers. And it is in that order because it's alphabetical and not because we value Ranganayaki's work any less. After almost 9 months of cataloging and processing, it is now in the shape that it is in today. And so today I'll be taking you through a quick overview of the collection, giving you a glimpse into the kind of materials present in the Krishna, Bahadur and Ranganayaki papers. The archives, for those who are new here, is a public collecting center for the history of science in India. We are a pretty young archive. We started in 2019, and since then we have been collecting, preserving, cataloging, digitizing and making available free of cost archival material related to the history of science in India from the past 200 years to the public. The archives has 4 main areas of focus. We engage in research work related to archival processing and oral history. We collect and preserve and make accessible private papers related to Indian science. We run an education outreach program for university students, high school students and teachers. We also conduct public outreach events such as this archives public lecture series. We also have an exhibition space and the archives are open to the public. We'd be happy to give you a tour of the space later today. Archives are also in a state of perpetual incompleteness. We have spent over 9 months cataloging and processing the material and making it available to all of you. But that is just a preliminary step. Every time a researcher or a user uses the collection and looks at the collection, it is their knowledge also coming into the archives, and we would love if you go through the collection and give us any inputs that you have in the metadata that we have created. We'd like to thank our funders and also the Krishna, Bahadur and Ranganayaki family who have very kindly donated the material to the archives. You can access the collection on our webpage, which look on the catalog that looks like this. This is how the Krishna, Bahadur and S. Ranganayaki papers appear on the catalog. All the information that you see here from content description, dates, creators, conditions governing access, use, organization are all collection metadata that the archivists have created. If you scroll through further, you can also find biographical and historical details about the people associated with the collection and also the collection itself. There are also languages represented in the collection. There is Bulgarian. If you want to search what is the material in Bulgarian, you can go to the catalog and search Bulgarian and you'll find the folder associated. Now, the collection is arranged into all these series based on the form of the material. So there's correspondence, administrations, research writings and outputs, research data and references, non-academic writings, personal and ephemera. Now, if you click into a folder, say from 1950-59, Embassy of the USSR, you will find scope and contents, that is what is in the object, the dates represented. So if you go and click on this digital file, then you will go into this digital viewer where you will find what is in that folder, that is the material in the folder. It takes you to the actual object and you can read through the documents right there in that browser. Now that we know how to access the collection, there is also a quick distinction that one should make. That is, accessing the collection is different from using the collection, and that requires separate permissions. However, Ila Bahadur, the donor of the collection, we are in discussion with her about assigning Creative Commons to some of the material in the collection, and that would make sharing and using these material easier. Now, before I move into giving you a quick glimpse into the collection, I do want to give a fair warning that this is going to be text heavy. So do not worry if you're not able to read through all the material that is being shown. It is all available as digital objects on the catalog and you can go through it in your own time. Now, let's go into the material in the collection. So, this is the first series, which is correspondent, and it's one of the most compelling series, I would say. And these letters reveal a constant engagement and with the global scientific community. You'll see exchanges with key figures like Alexander Oparin that was mentioned in the talk before, with Carl Sagan. There is also correspondence with John Aforo, who was also mentioned in the previous talk, I think. With John Aforo is inviting Bahadur to be a part of his lab. Neuberger is writing to Krishna Bahadur saying that his early skepticism of Bahadur's work was unjustified. But there is also disagreements about the work that is visible in the collection from Sydney folks. But then there is also extensive correspondence regarding the International Symposium on Origin of Life. He couldn't attend the symposium due to multiple issues, which included funding issues, I think, visa issues, etc. But also, a part of this Symposium on Origin of Life is airline documentation. That is, he's writing back and forth with airlines, asking for the tickets to be booked. It's like pre-make my trip, pre-is my trip era. Now, there's also correspondence related to Gordon Research Conference on Origin of Life. Adolf Smith is writing to him to be part of the conference. Now, there is also letters during the emergency era, which shows how science was being done during turbulent times. Moving on to another series, which is the research data. This includes lab notes, pH measurements, chromatographic analysis, graphs, etc. And a big thanks to Nayan Chakrabarti, I think he's here somewhere, who has helped us with making sense of the scientific data. I'm not from a scientific background, so it is difficult for me to make sense of the scientific data. So Nayan kindly helped us with understanding what the data is, and thank you so much for that. The value of the series is also, I think, is in its granularity. It contains daily observations logs, pH tables, and probably you can see how a wiggly line on a graph may become an evidence of life. Now, there's also Jeewanu, the microscoping images of the particles themselves. There is scanning electron microscopy images, a lot of microscopic images of the particles. Coacervids, I think. We also have administrative documents, which include details about their professional trajectories and denial for promotions, even though they had seniorities, which are all present in the collection administration series. Then there are brochures, newsletters, meeting updates, a lot of public documents, public-facing documents from the ISSOL. So it shows the kind of international organizations that they were a part of. The archives also holds records from the University of Allahabad, in which they were a part of. There are grant renewal documents, no objection certificates, viva voice documents, appointment orders, and it kind of captures the administrative weight of running a lab too. Now, this is one of my favorite objects, I would say. Before we had reference managers like Sotero or Mendeley, there was this, a meticulously handwritten database of references. I call it the pre-modern Sotero. Now, we also have drafts of writings, draft of mostly Krishna Bahadur's writings. There is his thesis report under Dr. Neel Ratan Dar in the University of Allahabad. There is report that he wrote after his Nuffield Fellowship at the Imperial College London. There are drafts of lectures that he gave on different symposiums, etc. Now, this is non-academic writings series, and this is the material that I think primarily grounds their work as teachers and scientific educators. There is, we found writings on Hindi, on cycloparaffins and alkanes ka nomenclature, so it's mostly in vernacular. And also there are comments, general remarks about a chemistry syllabus. And another fascinating object was this material. It is titled Prakash Se Pani Tuta. I think it literally translates to light breaks the water, and I think it's a take on photolysis, but if any of you think it's something else, you can scan the code and you can go to the digital object, and if you think it's something else, please let us know after the presentation. If everyone's done scanning, let's move on to the next slide. There are photographic material similar to the other slide. So if you know who are present in this group photographs, please come and let us know after the presentation. If some of you are here also, that will be great. Yes. Oh, that's nice. It's like a full circle moment. There are also stamps, so it kind of gives a glimpse into the personal hobbies that Krishna Bahadur had kept. These are stamps from the different letters that he received and which he has curated. Also now, a critical gap that we must acknowledge is that all the material that we have been showing are mostly Krishna Bahadur's material, and we mostly have Krishna Bahadur's material at the archives. All we have from S. Ranganayaki are these material, which include certificates, biodata, list of publication, some of her lab notes. This was titled Culture of Feast, Notes on Experiments. But there is also this in between all her lab notes, which shows the life in the lab. It strangely humanizes the person. But we are in communication with the family and friends so that we get more material from Ranganayaki in the archives. Now I'll end with this. This is a correspondence that was sent to Krishna Bahadur. It is not in our archives. It is in the Welcome collection. And it was sent to Leslie Orgel. And we have the letter that Leslie Orgel has sent back to Krishna Bahadur in the collection. So it's that no archives is exhaustive. Only if you read all archives together will you get the complete story. Finally, a story about how it reached the archives. At the archives, when it reached, it was in this state, and Anjali, our archivist, found a surprise guest, which was gamaxine. And for those who don't know, gamaxine is a pesticide now banned globally. So it was the preservative of choice back then. So Anjali, Sindhu, and Kinjal, who are our conservators, donned their protective gear and cleaned every single sheet. And so you could say this project involved quite a lot of toxic labor, maybe. So it does truly take a village. I thank all the archivists and conservators who cleaned, catalogued, and preserved the history. And also a special shout out to the Archives 2025-2026 team. Thank you so much. And this is the QR for the collection. You can go to the QR, scan the QR, and find the catalog there. Thank you. And now I'll hand it over to Dr. Shashi Thutupalli, who will go into more details about the science and the history of the collection. Thank you so much.
Okay, thank you. Thank you all for coming. And today is a celebration. Sorry? Oh, today is a celebration. It is the 100th birthday of Krishna Bahadur to the day. And in fact, it's a very happy thing that we're also joined by the family. And I think we also remember Chandran, the brother. We have a seat for him today. And I think I want to say that these are two remarkable scientists, and I don't use this word lightly. Also a remarkable family, because they have somehow realized that this is important stuff and have kept this for all these years and has made today possible. And I think all of us are showing this picture in our talks because I find it to be a remarkable one. And I think it, at least to me, it gives a very nice sense of the kind of scientists they were. And in fact, just yesterday, I heard a most remarkable story from Dr. Vinod Gupta, one of the students, when Ranganayaki had surgery and she was in hospital, and Krishna Bahadur was accompanying Ranganayaki in hospital. And of course, they were there most of the time, and the students and the family were going back and forth between home, lab, and here. And these two are very curious and passionate scientists, and they want to know what's going on in the lab. So much so that the students actually set up their experiments in the hospital, right? While cycling down to the hospital without brakes on their cycle and so on. So it tells you something about the kind of scientists they were, right? So what I want to do today is to tell you a story, my own story actually, in where I reached this and so on, and some of the science that we're doing behind it. And that story starts with Obaid Siddiqui, because many of you here may have been probably here at NCBS for the first time. Obaid Siddiqui, molecular geneticist, was brought in by Homi Bhabha to start biology at the TIFR. Over the years, that effort grew. And here's a very interesting connection back to Vidyanand Nanjundiah, who gave our opening public lecture, where together with Obaid Siddiqui, Vidyanand Nanjundiah has proposed a new center for biology in India. These are sort of the roots of where we are sitting today. And they call in this proposal that biology should be integrated with physics, chemistry, engineering, mathematics, and so on. And even there's a mention in that report of some theoretical work going on at TIFR on the origin of life and things like that. So that's Vidya back in the day. That's where we are today, thanks to some of these pioneering efforts. And within the NCBS itself, there has been this set of people that have, I want to say for now, for close to 2 decades, been engaging with this interdisciplinary aspect, particularly the physics and the math and so on. And for the last 13 years, we've had this so-called Simon Center for the Study of Living Machines. This was already in place when I arrived at NCBS. We started doing various sorts of things in the lab. But one of the things we started doing was to start discussing with Sandeep Krishna, who you saw also in the previous picture, about the question of the origin of life. And Sanjay, I hope, is online on Zoom. So we had this meeting, Sanjay, Philippe, a collaborator of Sandeep and Sanjay, and also mine now. Yoshia was a postdoc who joined us from Japan. And Sandeep Ameta, who's sitting here, joined us as a senior postdoc in the group. Nayan had just joined our group as a PhD student. And we were all having this conversation about origins of life and what we could be doing. And I was mostly following the conversation in this half of the room because it was all about the kind of language that I'm used to from physics and network theory and so on and so forth. But here were coming comments from chemistry. And I had absolutely no idea about chemistry. And I hated chemistry in school. And I said to myself, well, if I really want to move forward in these conversations and also do something, I really must learn chemistry. And so I really started reading up. And we have heard much of all of this history in Vidya's talk, right? And I won't go over those details again, but this was some of the things that I was reading up and trying to understand what actually might go on in this kind of setup and things like that. Just one comment I want to make here, perhaps, which relates to Sidney Fox and the work of Miller. The work of Miller, as Vidya said, involved this gaseous chamber, which was electrified, gave rise to amino acids. Sidney Fox, and primarily his postdoc Harada, started with the question of saying, we have amino acids, what can next happen? Right? And so they started doing all kinds of treatments to these amino acid mixtures. And one of the things that was successful for them was heating these samples to 700 degrees and so on, which is why I think he started thinking about these volcanic conditions where these sorts of things might be going on. And what he found was that these amino acids were polymerizing at these very high temperatures and when they were being cooled down. And when they were cooled down, these polymer mixtures were phase separating into what they called microspheres, which were comprised of these proteinoids. They were really trying to understand these as the coacervates that Oparin and Haldane thought about, and trying to induce more and more biological or cell-like features into those objects. Another book that I read during this time was that of a Hungarian chemist called Tibor Ganti. And Ganti is known for what he called a chemoton, a chemical automaton, where he proposed that a metabolic or a chemical reaction network could couple to a compartment-forming network, and that he imagined to be a protocell, right? And in his book, This Principles of Life, he was talking about possibilities of experimental systems which relate to this idea of a chemoton, and he describes the work of Sidney Fox, which I just discussed to you. He is slightly critical of all of that stuff. He's saying that our system of criteria is not satisfied by Fox's microspheres since they do not metabolize. He actually even notes that, in fact, Fox's original aim was not to synthesize living systems, but to investigate the possibility of abiotic production of protein-like compounds, and that he was actually trying to push them, and in Bahadur's words, actually, by alchemic means, to make them lifelike, right? But rather instead of a more principled way of doing this. And following these paragraphs in the book of Tibor Ganti was this one, which said the other scientist who has performed direct experiments involving immediate biogenesis is Krishna Bahadur from India. Bahadur and his wife Ranganayaki have been working on the spontaneous formation of microspherules containing molybdenum atoms from primordial-like soups. This is striking. Their experimental research has not attracted much attention in literature, although their results are the most interesting and promising in the field. And this caught my eye. And it actually goes on to say that they're getting these particles, which they call Jeewanu, particles of life. They have metabolic activity, a photosynthetic network, but also membranes and so on and so forth. And I hadn't heard these people's names, and I said I have to Google a little bit to find out. And I actually found these pictures of them on Google, of Krishna Bahadur and Ranganayaki on a website called Jeewanu.com. This website was actually maintained by the family. Although the phone number listed there, I believe, was an incorrect one. We never were able to reach that phone number but these are the pictures that I found. There was some information about the Jeewanu, and I started digging further. And 1953 was when Miller published their work in the journal Science. In 1954, Krishna Bahadur has a paper in Nature where he makes amino acids using sunlight as a source of energy, no spark of lightning. He starts with formaldehyde and potassium nitrate. Potassium nitrate is in the mixture. Formaldehyde is by now thought to have come up fairly early in the scheme of things, so it's legitimate to use that. And his motivation here, as he writes in this paper at least, is that plants are able to synthesize proteins and amino acids using this method. Can I, as a chemist, do this? And indeed, that's what he announces in this paper, that he's been able to synthesize amino acids from formaldehyde and potassium nitrate. And in 1958 is a huge leap, in my opinion, where they remove the potassium nitrate from solution. Now he notes that microbes are able to fix atmospheric nitrogen. And they do that by using molybdenum as a catalyst. And so he says, can I use molybdenum as a catalyst in my solutions and fix atmospheric nitrogen to make amino acids? And this paper announces exactly that, fixing atmospheric nitrogen to make amino acids. And it is here that I can find at least the first connection to the origin of life question. He says the possibility of achieving amino acid synthesis without the help of microorganisms has involved some aspects of the problem of the origin of life. And then he cites Oparin and so on in their thinking, also Miller. This work, people have paid attention to it. Miller and Urey synthesize, at least up to 1959, many of the advances in the field. And this work of Bahadur, these two papers, have indeed found mention here. As we have heard before, Oparin organized what was the first symposium on the origins of life in 1957. And I've just put a smattering of faces to the names. The who's who, in some sense, were on the invitee list. And from India are Professor Bahadur and Guha, the biochemist. And this I did not discover then, but this is thanks to the archives that I have this. I'm just telling you the story at the moment. And it was in 1963, 64, when this very bold announcement came of these so-called particles of life, Jeewanu. And I believe that from 58 onwards, they've been working on this chemical recipe. And they announce that these Jeewanu, which are particles a few microns in diameter, are compartments which metabolize, forming various kinds of biomolecules that they grow and that they bud, giving rise to new Jeewanu and thereby self-replication. And this was the long list of chemicals that I could find in the very first paper that I came across on this matter, right? Formaldehyde and a bunch of salts, inorganic salts. Another thing that I found was this picture, and I'll come to the provenance of that picture in just a moment. But there was also a review produced by Linda Karen and Cyril Ponnamperuma, who were at NASA. Ponnamperuma had just set up the exobiology group at NASA. And they reviewed this work on Jeewanu. And I'm not going to go into all the details of what happened and so on and so forth. I will leave it for you to dig into the archive, because it's a very fascinating story of what happened between then to here. But essentially, this NASA report questioned the sterility of the mixtures, suggesting that these could be contamination. And they also said that the description of the experiments was inadequate. and therefore it makes reproduction of these experiments very hard. Right? And this was about the information that we obtained when we started this. To me, it was so the provenance of this picture is from this article written by a historian of science, Matthias Grote, who I also hope is online listening to this. So he wrote at least a little article, and then you will again see Vidyanand Nanjundiah here, because this is now published in the Journal of Biosciences. And this is an article which recounts, at least to the extent that Matthias could dig up into the history and the broad perspective of this problem. Right. I will already tell you some content of these papers. Right? In fact, this is a series of 3 papers. The first one is what they call a theoretical outline of the approach to the problem. Right? And I find this especially striking, apart from how they have designed their chemistries. I will come to some aspects of that as I discuss what we have done. They take a significant deviation from what has been happening at the time, which is, the focus has really been on trying to recreate conditions which were thought to be prebiotic on Earth, and put that into the lab and ask, how can we recreate this origin? Bahadur actually decides to do away with that completely. He says the logical approach to this problem is to take into account those properties of living systems which are common to most of living systems of today as necessary properties, and to consider how the components which are present in the present day cells were formed abiogenically, and in this, first giving preference to only those conditions which are even now present on Earth. If these conditions fail to give the desired results, other conditions which may have existed may be considered. But these conditions must be taken as second choice. He essentially said that what if we are wrong about this guess, all our efforts will be wasted. And as it turns out, much of our understanding of prebiotic conditions is quite different from what people had assumed back in the day. Right? And taken more broadly, they're asking here the in-principle question, right? And that is that's quite remarkable in my opinion. And they define also what they're going to go after: a system which is capable of growth, multiplication, and metabolic activity shall be considered as a living system, and that's what we're going to go after. Right? So they've set themselves that goal, and that's what they're going after. So this is broadly what we had. We didn't have the full recipe. We had that long list of chemicals, and here came Nayan, who had joined me as a PhD student in 2018, I think, and he had been working on some other problem. I gave all of this stuff to Nayan, and every so often we would talk about this. We would notice some patterns in the list of chemicals that, hey, it includes CHNOPS, it includes a catalyst, and this kind of stuff. And beyond those discussions, I was doing my thing, and as far as I was concerned, Nayan was doing his thing. But what he was really doing was spending nights and weekends trying thousands of reaction conditions. Right? He was spanning large chemical spaces, not blindly. He's a chemist by training, and therefore is able to bring that intuition into the game. And what he is looking for was that, when I first saw that electron micrograph image, these were spheres. And it struck me that why are there spheres coming from a solution of salts? This is odd. And if that's indeed contamination, I saw formaldehyde in the list. And I said, wow, that's an interesting contamination. If it's contamination, I want my hands on that contamination, right? And the fact that these were spheres was what Nayan was looking for in all his trials. Try something, precipitate crystal, no. And then one day, early 2000, he announced to me, Shashi, we are Jeewanu in the lab. So what did he have? He had these beautiful blue spheres. right? So he hit a regime where he was very quickly getting the formation of these micron-sized beautiful spheres. And I had to just run to the lab to see this with my own eyes. And the moment I saw this, a few things popped out at me. I mean, it's so uniform in size, right? This was, and the fact that there were also these contact points which looked like some signs of surface tension or deformability, liquid-like nature, and so on. And I said, hey, this is exciting and interesting. And right around this time, Janvi Halke, who was setting up the science gallery, came to me and said, Shashi, we are going to start off a program based on carbon. Can you build for us the Miller-Urey setup to show in this, to show as part of this thing? And I said, Janvi, we have one better than that for you. So we just set up this Jeewanu mixture. This was in the Indiranagar metro train station. And our intention at that time was the fact that we had stumbled upon something very interesting. We had redone this. This was a story. By then, I had been talking to many colleagues about this. No one had heard of these people's names. And I said, hey, this is a great story to be just told. This is a remarkable, as Vidya said, science in India story. Right? So we set this thing up, and then it has moved to the science gallery. There's a version of it outside which you can look at, and so on. And my talk is sort of deliberately braided like this because this is how it was happening, right? And as this thing was being set up in the gallery, and people were visiting, talking to us, and so on, experiments were proceeding. And looking back, this was the Jeewanu experiment redux, right? So, and we were throwing various of methods and our thinking into this problem. This had started really to consume us in a deep way. So the spheres are blue because they're full of molybdenum, right? So we subjected these spheres to spectroscopy and found these elemental signatures. Molybdenum is heavily enriched in these spheres. In fact, it's orders of magnitude enriched. And what is interesting is that the incorporation of these atoms into these spheres is non-stoichiometric. So you start with a certain stoichiometry in the initial solution, and it's a completely different ratio in the spheres, suggesting that bonds are breaking, new bonds are forming. There is indeed chemistry which is giving rise to these spheres. It's not simple precipitation, right? And at this point, we started to think, hey, you know what, colloidal synthesis is also something like this, and why are these not simply just colloids? And I dug myself into colloidal synthesis literature and found that there is actually no recipe for making colloids if you don't already start with a complex polymer, a long enough polymer which can nucleate, seed, and then grow, right? So even from that perspective, this was quite interesting and novel. Mukund asked at lunch, why molybdenum? So I thought I'll just put a quick slide. And because there's this full of molybdenum, right? Of course, the original aim of Bahadur was because molybdenum plays a prominent role as a catalyst in nitrogen fixation, right? But more importantly, and molybdenum, in fact, has been thought of people like Steve Bennett, et cetera, to play a very important role in the origin of life. And the argument goes that molybdenum has this dynamic valency such that it can actually participate in a wide variety of redox couplings and give rise to organic synthesis, right? So that's one of the reasons highlighted for its versatility as a catalyst in doing organic reactions. And molybdates in just simple water have the propensity for facile self-assembly. Right? In fact, it had been a long-standing puzzle that some lakes in the US were very blue, what Native Americans called blue water, and it was a mystery of why this was the case. And in the early 90s, this puzzle was solved because these waters were full of molybdenum, and they were assembling into these nanostructures, which then resulted in the blue color. Right? So molybdenum has this ready feature of also giving rise to structure. Right? So the spheres that we were forming are compartments. Right? So here you can see an SEM image where you see a hollow interior with a very clear wall. A TEM image reveals the fact that the interior of this compartment is not homogeneous, but itself is comprised of small little compartments, and the contrast over here is coming because of electron density, suggesting also that there are very sharp electron density barriers, and if these are mobile charges, then we have a battery there which can drive reactions. These are soft, so when two of them are contacting each other, they deform. We put them under an AFM and push them to see that they're indeed very soft and very, very sticky. The liquid is lumen-like, so we can do something like an osmotic shock and get them to crumple. We can clarify them using chemical means to reveal an interior. We can go in with a laser to disrupt the wall, and the innards, like this, the fluid just sort of bursts out, and you can see these little particles coming out, revealing their liquid-like nature. And these compartments grow. Right? So this is a movie over 8 hours, and you see that these exhibit very clean and characteristic growth. And you will also notice that even though they start off at vastly different sizes, towards the end they more or less reach the same size. So there is indeed a size-dependent growth rate of these guys, suggesting again that there is some surface catalysis-driven growth of these objects. The number of compartments also grows. This could be the fact that we could be tricking ourselves, because when we look here, these particles are heavy and they just rain down, and therefore it might seem like even though there are the same number of molecules above, more appear at the floor as time goes on. So what we did was to collect all of them and just look at how the total mass of these objects changed as a function of time. And there is indeed this sort of superlinear increase at early times, and even growing very slowly at past times. And I want to tell you that the recipe that we have of these 4 ingredients is minimal, in the sense that you remove any one of the components and nothing happens. Right? So it's nice to be able to have that so that we can tweak various things and understand mechanistics. This was happening, and again, I was discussing this work with various colleagues. At ICTS, we have this thing called Chat with Chart, where we give chalkboard talks of ideas that are occupying us, and I gave this Chat with Chart about this work. Deepak was sitting there, and there was also another occasion where Vidyanand Nanjundiah was there, and we were all sitting over lunch. And this inspired Deepak to get in touch with some of his old connections in Allahabad, and he one day sent me a phone number saying that he thought that Ila Bahadur was Chandran, the son's daughter, in other words, the granddaughter, and he gave me this number, and I immediately called Ila, and only to realize that she was the daughter, not the granddaughter. And of course, I was very excited, and I was sitting in this car actually to go meet the group, because we just had a paper come out and we were having a celebratory dinner. And I was just speaking faster and faster in the car, and somewhere in this conversation, she said she lives in Bangalore. And this was fantastic. She had just moved to Bangalore, actually. She was at ITI. And I just brought her over to the lab a few days later. And the moment she walked in, and she's an engineer by training, and the moment she walks into my lab, she asks, do you guys have a gas chromatograph? Do you guys work on coacervates? And what about RNA and all this stuff? And I said, how do you know all this? And she said, I mean, this was everyday life growing up. There was no escape from conversations of this kind, right? Yeah, even now it's amazing to see how many of the students she knows, and this is quite fantastic, in fact. Okay, and that's really how, so she came, she saw the archives and was very impressed with how the archives is run and so on. And thanks to that, we have the papers here and we have the launch today. So while all of that was happening, you know, the gallery effort was increasing. You know, they also visited, Ila and Mridula visited the gallery, see the exhibit. We are running workshops. In fact, now that we had access to these papers, I started realizing all the kinds of questions that were being asked of this work about its reproducibility and so on. And we decided that we're just going to bring in school kids one day to the archive, to the gallery and have them do this experiment. And pretty much everyone was able to make these Jeewanu. It's actually a fairly simple thing. In fact, the little girl standing here is the daughter of my friend. And these guys are board game designers. They design board games for a living, and they saw an interesting story here and started designing a board game. I think one of the I mean, these are outside, so you can see that later. So all of this is going on in the background as well, right? So we are testing these board games for them. We are engaging with the gallery and so on, and we are also updating the gallery with the science that's going on in the lab. So what's going on in the lab? We are finding out that these spheres, as they are growing, they are incorporating elements in different ways, right? So while the absolute amounts of all of the elements is increasing, even exponentially, as a function of time, the relative amounts, some are getting enriched, some are getting depleted. And on the time scale of about 8 hours, it seems to reach something like a steady composition of these elements. Carbon seems to continue to increase, right? But what we found, and accompanied by all this, the outside solution is acidifying, again suggesting of breaking of bonds, chemistry going on, and so forth. And this dissipation continues for months. So we put this reaction mixture into a calorimeter, and what we find is that the experiment just continues to produce heat for a month, 2 months. And it struck us that at that moment that when we put this into the calorimeter, there is absolutely no light. It's dark inside the calorimeter. And so we just repeated all our experiments in the dark as well as we could. The particles form, you know, everything that I showed you so far actually happens in the dark. And it turns out, and as I've learned also later in looking at these recipes and so on, so we are operating in a slightly different pH regime. We have a very highly acidic regime in which things are just getting kick-started. And it turns out that this photochemistry seems to be adjusting the pH of the system down here where everything can kick-start. So just having this abundance of protons seems to provide at least the initial kick for structure formation. And once you have structure formation, presumably because of the fact that you have all these gradients in there, that can sustain further synthesis. And because this was happening in the dark, this was happening in ambient light, and we had also set up an experiment outside the lab, literally in the Bangalore sun. And thankfully in 2024, there was no rains for nearly 3 months early on. And so we had an uninterrupted slot of sunlight to do these experiments. But we realized that none of these I can actually write in a paper and say, hey, reproduce these, because they have to come to Bangalore to do this and hope for no rain. So then we decided to actually set up a controlled condition in a clean room with a standard temperature, with a lamp that mimics the entire solar spectrum. And into this experiment, we actually threw in formaldehyde, which contains 13C, the heavy isotope, so that we could actually track the synthesis from the source formaldehyde itself. And indeed, there is direct organic synthesis from formaldehyde. So already within 15 minutes of making this mixture, there are many other compounds. What I'm showing you here are chemical shifts from NMR. So you get many peaks corresponding to a few organic molecules, but by day 21, that has complexified significantly. Formaldehyde gets consumed, new products get formed. To cut a long story short, we can analyze these using mass spectroscopy now. And what are being formed here are core among various other organic molecules, core biomolecular classes. So there are molecules which are lipid-like, amino acid-like, and what have you, right? So there is indeed this organic synthetic engine that's operating and giving rise to chemical complexification. And in 2024, this paper literally hit me out of the blue. And this paper was written by a bunch of ecologists. And they were studying sponges in the ocean. And they reported that these sponges, Thionella conica, were super blue because they were full of molybdenum. And they looked inside, and what they saw was that these were blue spherical objects, roughly the size of a few microns. They were shells. They called them blue vacuoles. They didn't know what they were. They said they may have some biological function. We don't know exactly what that function is. And they had also done the exact same spectroscopic analysis at what we had done. And it's quite remarkably similar to what we were finding. And some of the structures that we get in the lab, so this is structures about 42 days after exposure under this sunlight, they look morphologically very similar to what's being seen in these sponges. They also commented in this paper about the fact that in addition to this blue stuff, they were getting these yellow aggregates, which they attributed to different oxidation states of molybdenum. And so we just tweaked our recipes a little bit, just close to where we were operating, and indeed we find all this yellow stuff there too, suggesting that there could be very similar chemistry going on in these settings. Finally, when we were breaking these things apart, you saw these tiny little particles coming out. And when we looked back at some of our old data where we had high intensity TM images, we were seeing that there were bigger spheres inside these spheres themselves, right? And we thought that perhaps this is growth inside these spheres. And so we just tracked the particles that were coming out, right? So we took a few of these, burst them open, and just watched them. And indeed, they also grow into spheres. And we have some evidence to suggest that they're indeed also compartments, right? And their chemical signatures are very similar to these ones which we formed initially. I'm not claiming that this is reproduction, but I'm saying that this could be a very interesting route to get to self-reproduction in this system. So all of this is out on a different archive just yesterday, so you can read our paper. And in light of Vidya's comments, physics is the only game that matters, right? And sort of the story has, because of the presence of the archives, come full circle in a very, very interesting way. I started the talk with Obaid, and we received all these papers into the archives from the family, and the archives folks are feverishly looking for any mention of Jeewanu in the archives, and they can't find anything, and they just typed in a different spelling, and they found this. So what is this? This is a talk given by Obaid in the All India Radio on the origin of life. And he has left a copy of that script, and the All India Radio people have returned this. And Obaid has given this talk in 1967. So he goes over most of the talk, outlining all these various ideas and discussions that are being that people are thinking about origin of life. He's discussed these coacervates of oparin and so on and so forth. And he says, it is not difficult to follow this line of argument further. Let me stop here. But he strikes it out, goes on, and he says, I have been talking as if origin of life is merely a problem for exercising imagination. This is a bit out of date. About 12 years ago, Miller designed a simple experiment. This is the famous Miller experiment. And he says, the organic building blocks of life are formed, but then how do they go on to form polymers and higher order aggregates, proteinoids, etc., is an open question. And he says, one of our own countrymen, has coined the term Jeewanu for just such entities. And these things are far from living, but they have interesting properties and make excellent models for investigating the beginning of life. And these various ways are open now to experimental investigation. So in some sense, it's very appropriate that this has come full circle, and we are continuing this work. I just want to make a couple of comments about this. It's quite remarkable that we had a time when a talk about the origin of life was on All India Radio. And it's also quite remarkable that there is recognition. You see, he has discussed Miller, and by this time, Fox and others who are known, in fact, to Obaid, are big names in the field. And he, in fact, recognizes the work for its worth. And I believe this is something that is not so much in vogue these days, unfortunately, where we are outsourcing some of our judgment to where it's published and what its impact factor is, and so on and so forth. And so I think rightfully, as also Vidya said, Bahadur and Ranganayaki belong in this pantheon. And that's where I want to end my talk. And if you have any questions, I'm happy to take them.
So you have compartments forming, you have something like reproduction. As you know, and as was mentioned in Vidyanand's talk, there's the informatic aspect, the precise reproduction of a particular combination. So is there any chance of doing that in this system?
Yeah, so at least so let me just go back to this data because it's interesting. This suggests that there is one kind of information being passed on, is the chemical composition, right? Whether that's sufficient to drive further diversification, complexification is indeed an open question. And I must also point out that this hasn't happened spontaneously. This is provoked because of our breaking it, right? And maybe all these features come together in some ways, right? And there are also ingredients for the formation of these information-rich polymers. There are nucleobases, there are sugars in there. Could that result in RNA? Hopefully time will tell. Yeah.
Yeah, so Shashi, these compartments that form, so I mean, the images sort of, it looks like there is some kind of an envelope that is formed first. Is that chemically distinct from what's inside? Have you analyzed that?
Yeah, so yeah, that's a good question. So we haven't yet been able to spatially analyze molecular composition. We have spatially analyzed atomic composition. Right? There is heterogeneity. But what we have done is to isolate the compartments from the supernatant and analyze those materials separately. And we find that organic molecules are significantly enriched in this compartment phase. Whether they're in the core, whether they're at the wall, this we don't know yet.
Is a comment in order, not a question?
Yes.
This is just a personal experience of mine. Sometime in 1976 or 1977, I forget the date, Professor Sharat Chandra and I, he's somewhere here.
He is here, yes,
so
right there.
I'll request him to add his recollections. We went to Hyderabad to attend an Origin of Life meeting. That had been organized by Pushpa Bhargava at the CCMB. And at that time, this interest in the origin of life had been triggered by the arrival of Cyril Ponnamperuma and his stay. He stayed for a while in Bangalore as well, as I recollect. without taking long, I just want to say there seemed to be a fairly large number of people who were interested in the question. As I recollect, most of them were chemists, and much of the activity, certainly some of the activity, was centered at the BARC, catalyzed by Dr. Chadha, who I think was in the chemistry group there, or was the head of the chemistry group. So I think it would be a matter of interest to see how interest in the origin of life has figured within India. It seemed to be quite active at that time.
And maybe I can just add to this comment a little bit, because we were having conversations with Vinod Gupta and some of the other students, and they indeed have interacted with Pushpa Bhargava and so on, and they have their thoughts, which perhaps they can share as the evening proceeds. Yeah. Yeah.
Just to follow on that, I think work like this can happen at a place like NCBS, but in many other places, this extreme focus on health and applications mean that work like this will not be encouraged. A young person will not decide to go forward. So we're privileged, but what do you advise, more generally, if you have curiosity and interest like this, how to do it under the radar, wherever you are?
I don't know. I don't know. I guess. Sorry?
Here we can.
Sorry. No, I think this place has really been set up well to stay under the radar, right? And the people I thanked in the very beginning were for that reason, that because that's what's enabled in a place like this, right? And I think it's very important to sort of keep these spaces and this way of working possible, right? If we go chasing after health and glory, maybe we'll really miss interesting things. Yeah.
Sorry, Shashi, just coming back to this picture.
Yeah.
So do you think there's chemical reactions that are ongoing, the internal pressure keeps increasing, and it could, over some time, generate cracks, and the whole thing would crack and explode? And
it's completely possible.
how long?
It's
can you measure the increase in pressure and then maybe predict what time?
So you can actually see in this image that there are these dimples forming on the surface because perhaps it's pushing out.
Right.
What we find in our experiments is that this wall hardens as a function of time.
Right.
And we think that is because of oxidation. And I don't know the exact mechanism of hardening, but if we prevent this oxidation, they do remain softer for longer.
Okay.
And under such conditions, can the kinds of things you're asking happen? To be investigated.
So I guess I have a related question. What determines the size of the spheres or whatever particles? And the sponges or whatever the particles they saw, and what you see in the lab, are they the same size?
They're the same size. Yeah, they're roughly the same size.
So what is it that determines that?
I don't know. This is a very good question. I think we have to get to the bottom of this. One thing we know is that they're hardening as a function of time. And does that lead to some kind of a kinetic arrest, which keeps the reactions going inside but prevents this thing from going further? I don't know. Right? But just to go back to also Madan's point, so you see that if we have indeed waited 42 days, that outer sheath seems to have gone off, and it becomes more bulbous, or this grape-like structure, right? Perhaps that is going on.
Dr. Nanjundiah's comments about that Hyderabad meeting, I remember 2 episodes. I have a copy of the proceedings of that meeting. I looked through that, the people who participated and so on, and I don't see Krishna Bahadur in that. When did he die? What year did he die?
1994.
Oh, then he was very much alive.
He was very much alive, very much active.
And one other interesting aspect of that meeting was that JBS Haldane's wife, JBS had died by then, lived in Hyderabad, within a few kilometers of CCMB, and she had not replied to Bhargava's invitation. So I was deputed to go and meet her and bring her for the meeting. Ovaid suggested this to me. So I went to her and, Helen, why don't you reply to? She felt that they have insulted me, she said. I said, how can they insult you? And she showed me a letter which probably was not typed by one of Pushpa's secretaries, because he had a very meticulous secretary, but somebody from the conference, and they spelled Haldane as Haldene. And she felt that this is, they don't even know my name properly. So she took that as the reason why she is not attending, and she, in fact, did not attend that meeting.
Oh, wow. Okay. You know, a chemist would take offense too, because yes.
Hello, Shashi. Very nice talk. Thank you very much. When I interacted with Dr. P.M. Bhargava, and we were in a meeting, so where did you publish? And you see, there may be a contamination like that. You should confirm and recreate those experiments. Repeatedly, he was suggesting me the same thing, because he was an Indian delegate to the conference, and being an elderly person, he was guiding me. You should recreate, and there may be contamination. Sir, I told him that we are using 1 volume formaldehyde. It comes to around 7% of the total mixture, and even 2% is sufficient to rule out any contamination. We are preparing our samples under aseptic condition. Sir, more than 50 students have done PhD and DSA also under Dr. Ranganayaki and Bahadur. Thousands of times each students have prepared these solutions on the variety of biochemicals and various processes they have studied. And repeatedly we are getting these results and trying to publish where possible, because we did not have sophisticated instruments in our lab. So this is how we are struggling since long time. And thank you, sir. Personally, I would like to ask you, what is your perception about the contamination in the mixture?
This is not contamination.
There is no contamination. Thank you.
No.
Thank you very much.
Yeah, of course. Yeah, I mean, I'm speaking to the source. No, so we have followed every care that it to work with aseptic conditions, but this experiment that we have done with 13C rules out any natural contamination that can come in this system.
I was working at Ultra Fast Center at Chennai with some laser flash photolysis experiments, and we could see that when we radiated the samples with laser, and there was photoproduct formation from 10 to the power minus 9 to 10 to the power minus 16 nanoseconds. See the photo, it is an ultra fast reaction. It's amazing. Even I could not find out the quantum yield of the mixture, because there were superfine particles were in the sample. There were so many aberrations. I worked for 6 months, but I could not calculate the quantum yield in the mixture.
Sure. No, I emphatically declare today that this is not contamination.
Thank you very much. Thank you. Thank you.
Okay. Yes.
How it look like over longer time incubation, whether Jeewanu should be stable, yeah, it should be changed in various different way.
Right.
And what are that lifetime of such type of molecule?
Right, that's a good question. I mean, so this sample is taken 42 days after, and you see that they're not those uniform perfect spheres that were formed initially. There are morphological changes that have gone on. There are other morphological changes that happen as a function of temperature and so on and so forth. And they form tube-like structures and things like that. Yeah.
And one query key in organic compound, what we learned from the basic textbook, the carbon hydrogen should be higher comparable to the other molecule, but here oxygen and molybdenum should be higher. How should define these two actual
that is extant biochemistry, right? So here we are talking about processes that might give rise to that chemistry, and that is a huge gulf, I think, in the origin of life question.
Okay, thank you.
Okay, so I think in the interest of time, I should stop here, but I want to end actually with this picture. This is from the retirement of Krishna Bahadur in 1986, and you see that they had a remarkable group, and also very fascinating to see how many women were part of this group, and yes, so we certainly have. Yes, we have Dr. Suman, we have Dr. Mridula Bahadur, we have Dr. Vivek Agarwal. Sir, you have to. This one. So we have the three. We have the Lal Sweater Wale. So we have the three of them here. And I would like to invite first Dr. Mridula Bahadur and then Dr. Vinod Kumar Gupta to say a few things about your time in Allahabad and some of your comments.
First, I want to tell you that I wrote this speech with my sister Ella. We together went back and forth, and I think she should be the ones reading it out here or saying it out here, because she is the one who has put in so much of effort towards making all this happen, and she's the one who met Dr. Shashi, and she got all the things here. But I think because I was coming from a longer distance, she is asking me to speak instead of. So I'm in Boston, so I came just for this event today. And it's a very emotional day for us to see all of you sitting here. It has been, our father died in 1994, and our mother passed away in 2002. And so many years after their death, today we are sitting here in this room discussing a body of work which was done in the 1950s. So it is 50s and 60s, and so it is remarkable. It's so difficult for us. It's very difficult to be here and to see all this so many years after. And trust me, as we were talking the other day, if it was not now, if it was after we were gone that this thing had come up, maybe we would not have been able to give you all the stuff that we have given. How long can you preserve something so fragile in a house that has termites and moisture? But our parents knew. See, they did not get the success they deserved in their lifetime. They had so much criticism. They had so much of difficulty. They had so much of funding problems. But they knew. They knew what they did. They knew the magnitude of their discovery. They knew it was worth preserving. They preserved everything. They preserved, you see how letters are preserved. So every single thing. I think my father was more meticulous with preserving his things than probably my mother was. That's why you're able to find more of his stuff than you are able to find hers. He was cataloging things. He was keeping them properly. And our mother used to tell us that keep this safe, because one day somebody is going to come. Somebody is going to come looking for this. So you make sure that this goes in the right hands. So in 2008, when I was visiting India, I was visiting my brother. He called me and my younger sister. My older sister was older, so she was not asked to do things. But the two of us were called. And we were told, put this properly, we have to do something this time. I don't know how long I'll be able to preserve all this. So he got those 2 large trunks that you saw, the metal ones, and she and I, we put them, and we are sorry we put it under so much of gamexin. We did not know what else to do to keep it from just getting eaten by the termites. So much of stuff has been thrown away from those shells. So in spite of all the criticism, in spite of all the difficulty, in spite of all the whatever problems they had, the question is, how did they stay on this? Because they believed in it. They really believed that this was work that will come into the limelight at some stage. And they had each other. They were very supportive of each other. They were very, so much of admiration for each other. I have seen my father, the way he used to admire my mother. It was remarkable to see. As we were growing up, every time we were told, listen to her, she is a very intelligent woman. Constantly there was praise for her, and the mutual admiration was very high. So probably that is what kept them going for so many years. You know, somebody was asking us that how come they met and married, Dr. Ranganayaki, Subramaniam Ranganayaki and Krishna Bahadur. It makes an odd combination in 1952. So yeah, it was not an easy marriage. My mother came to do her PhD in Allahabad from, I'm talking personal things. I'm not talking science because everybody has said so much science. So I thought maybe I should give you a glimpse of the personal side of it. So she came to do her PhD. And he had done his PhD from the same lab from Dr. Dhar, and they were adjacent tables. They worked at adjacent tables, and my father saw the very intelligent woman, and he knew that he cannot let her go. So that's how they got married. And my father had a very difficult childhood. His father passed away when he was in 10th grade. He had to give tuitions to earn money for his education. And luckily for him, there were many good people around him. They helped him financially, they helped him emotionally, they gave him, supported him. My mother had a little easier start. She was the daughter of an English professor, so she didn't have to go through that kind of emotional hardship, financial hardship. But she was an extremely intelligent woman. She spoke multiple Indian languages. No accent. You could not tell from her Hindi. She didn't grow up in North India. She grew up in South India. You could not tell that she was not a North Indian. She spoke perfect Hindi. She could translate German and Russian papers into English. Anytime, anything I wanted to ask, I could just go to her, and she was like Google. I'm not joking. She was like Google. The only thing that comes anywhere near her is Google. When she passed away, I'm just not kidding. That's how I used to feel. When Google came, I said, oh, this is the closest thing I see to her. I could just ask her anything. She'd first scold me, and then she'll tell me. So it was remarkable growing up in that household. They thought science, they talked science, every conversation in the house was science. The PhD students used to be in and out constantly. There was so much I wrote, but I've just gone off script. So it was, the research students, the PhD students were all intermixed. Every experiment was discussed together. I never saw them argue over results. I never saw them argue over experiments. They were always working collaboratively. It was a very good collaboration they had. And what else did I want to say? I had so much here. So I think at this moment I would like to take this opportunity to thank all of you. To thank all of you who have shown up today, to thank the archive for preserving all of this. And I want to thank ILA for having made sure that everything came to this place. I mean, Murli Madhavan and ILA, they have worked relentlessly to get everything to this archive. My sister who has come with her ill health, she's sitting here. We left a seat for our brother who passed away, who doesn't even know that any of this is happening. So we left him an empty seat over there. And we have to thank all of you. Dr. Shashi, I just cannot even thank you enough for what you have done for us. To pick this up, and to bring it into the limelight again. And all of you who have spoken so highly of him, and it's just overwhelmed. And the students who have come, look at them, they have traveled all this distance to be here, to participate in our happiness today. So, Ila, do you want to add something? I'm forgetting what I wrote also. So we are very grateful to all of you and for bringing this forward, and to Dr. Vidya for having spoken and to bring this up forward to us. We are really grateful today. And I think I'll stop now because I'm just overwhelmed. So thank you. Thank you. I do this every day for a living, and look at me again.
Can I invite Dr. Vinod Gupta to share a few of your perspective as a student?
Namaste. Great. It was really an academic feast for us. Big thank you, NCBS. It is a profound honor for me and all our members from the lab to stand here at the occasion of centenary celebration of Professor Krishna Bahadur and the launch of the wonderful archives, so that everything was put in the proper perspective. Love is the driving force of life. You have seen when the Mridula was speaking, what she was communicating. That is the essence of the life. This is what the Professor Bahadur and Ranganayaki, we have become a family. We went there to join PhD, but still throughout my life, I am working on the same problem. No difficulties. Wherever we went, we had collaborations, people encouraged, and every lab I had a table to work. So this was the potentiality of the work. Wonderful. And I must add that establishment of this archive is really, it is in tune with the most prestigious institution of the nation. It is with vision and mission, and they have archived a problem where it will be accessible for the global audience. All scientific scholars will be able to access. Professor Vidyanand ji, learned professor, wonderful talk. I don't have words for the dedicated efforts made by Shashi ji and his wonderful team, starting from hospitality, warm hospitality, very touching hospitality. Everything they are taking care of. That elder sister is having problem, everybody is well taken care of. Wonderful. So thank you very much, Shashi ji, and thank you for the Chandran honor. And it was very auspicious beginning for this work. And I just want to say a few words. Professor Bahadur used to say that all energies can be traced to sun. And there is a Mahasuri mantra. Professor Bahadur, he was the true product of Indian culture. I must say there is a Mahasuri mantra. Nunam Janah Suryan Pursuta. Sun is giver of life. Life originated in water by interplay of primary elements. In Rig Veda also there is a Nasadi Sutra, which also says very important findings about the origins of life. It's wonderful. We are using weakest source of energy. Solar energy, we are using visible spectrum and whatever it is. We are using sunlight. There are 25 elements in nature according to Indian philosophy, and 25th is the Purush, sun, giver of life. So the message came from this Indian knowledge system. We have derived, we are inspired by this ancient knowledge. It is an Indian knowledge. It is one of the most ancient culture of the world. So we are having, and we have, I simply want to sum it, that why Jeewanu in 21st century? This is an open era of innovation. Let us share information, let us collaborate, and let us reveal this Indian wisdom as the global one. So inspiration is the love of the family, togetherness of the research group. It's wonderful. So I think this was not an occasion for honoring of just a scientist, but a question that how did life came into existence? How this lifeless matter was transformed into living system? What were those physico-chemical conditions? When in 60s, when the people were busy, the scientific community was busy with the space researches, stardust and the soil samples and all those things, Professor Bahadur and Ranganayaki, they were investigating the simple reaction vessels in their small lab. It was wonderful. So, this couple, Ranganayaki and Bahadur, they created self-organizing, metabolically active protocell-like structure named Jeewanu. Jeewanu is a Sanskrit word for the particles of life. We have not synthesized life, but we have synthesized a system which is comparable to life. They multiply, and when there is some synthesis, they grow in size, and they show metabolic activities. The fascinating result have been demonstrated by Shashi ji. Wonderful work. So we have all simple inorganic, organic structures, hybrid structures created by simple presence of in aqueous solution using some inorganic, organic materials. If there is in Vedic literature, it has been said that if there is any magic on the earth, it is contained in waters. Bahadur sir, he used to tell us, once Professor Briggs, when he heard about his experiment, he came to him and he said, Bahadur, if you have synthesized peptides in aqueous solution, then half of the problem of origin of life is solved. We have said that there are peptides, people are not believing because there may be some contamination like that. So further, it is in scientific experimentation. Again, we will do again and again, we will try and test those hypotheses. We are getting small monomers of all, diversity of monomers are there. I think Professor Shashi has just started, but he has very proper plan to work on these systems. And in last days, Professor Bahadur, in collaboration with NASA Ames, and when he was studying the functional properties catalyzed by these Jeewanu, and he could see that under certain specific condition, these Jeewanu can show the photolytic decomposition of water utilizing sunlight as a source of energy. So to test this idea, further they used N15 and C14 radioactive tracer and D2O also labeled systems were used, and they could confirm that for fixation of nitrogen and carbon dioxide, hydrogen is coming by photolytic decomposition of water. So Kyan Smith was very much impressed, and he has written a book called Genetic Takeover, and he has published by Pergamon Press, maybe having, Shashi ji must be having in his library, that some earliest energy transducing systems were similar to the grade of Jeewanu, which may had some inorganic crystal genes to catalyze the photochemical fixation of nitrogen and carbon dioxide. So these are the various catalytic properties are there, and we are having abiogenesis. This is the age of synthetic biology. Dr. Bahadur started synthetic biology in 60s, and he got these things. Now, lot of possibilities there. Synthetic biology is there, artificial photosynthesis is there, hydrogen production is there, sensor development is there, so many simulation and abiogenesis, a lot of significant biogenic molecules are synthesized in the mixture. So let us work. So I will not take much time, the meeting has gotten long, but so now the work is going on, like, can we build an artificial cell? How does the information arise from chemistry? Could life emerge from elsewhere in the universe? And where lies the boundary between the living and non-living? So the Jeewanu, the launch of this Jeewanu digital repository, we are not simply preserving the history, we are, the spirit of Jeewanu aims to learn about the chemistry of life, the emergence of complexity, and the boundary of consciousness and the unity of matter. So Professor Bahadur reminded us that science does not only pursuit of answers, it is the courage to pursue the mystery. So let us follow our quest and find out the bigger answers which these people, the path they have shown us. So thank you very much. We are already late, and so kind of you for giving us a patient hearing, and for everything was given so affectionately to us. We are highly obliged for your warm hospitality, and thank you very much. I very much touched by the family members who have, so many people, they have come from the different parts of the nation and the abroad also. So thank you very much for taking so much trouble. So kind of you. Namaste. Now this is going to be a new start for us. Thank you very much. Namaste.
Thank you so much for everyone joining us today. To everyone who raised your hands for not having visited archives, this is your chance. We have an archives tour happening for another 30 minutes. So please come join us at archives, which is in the next building, basement. There are arrows which direct you towards archives, and we also have a lot of archivists helping around. So thank you again for joining.